So neuropentraxin-2 has gathered quite a lot of momentum as being a more global biomarker for synaptic damage or neuronal damage in dementia. So it's not exclusive to Alzheimer's disease. It's actually been shown to be reduced in a number of different disorders. So the aim of our work here is really to develop a highly accessible immunoassay for this protein, which currently is only measured by mass spectrometry methods. So this is a method that makes it more globally accessible to researchers and maybe in the future clinical use. We have really good biomarkers to diagnose Alzheimer's disease, meaning that we can understand the pathology that's causing dementia, like phosphorylated tau, A-beta. What we don't really have tools for is to say how far along are you in that process, how much neurodegeneration, how much damage has been done. And the research prior to what we've done is that neuropentraxin seems to be a leading biomarker of this. And what we've done is been able to create an assay, which means that more researchers could be able to apply this biomarker to their cohorts and really address the issue of prognosis rather than diagnosis. Neuropentraxin is a biomarker that decreases as disease progression. So as I previously mentioned, that we needed to have sensitivity in order to detect neuropentraxin in advanced disease stages. We also tried to validate this biomarker in blood, and this is seemed to be very, very challenging. So we used the assay to measure blood samples that had paired CSF, and we knew this assay was working well in CSF, but had greater challenges in plasma. So in our paper, we showed that neuropentraxin was actually increased in plasma, but we also showed the evidence that this was a hard biomarker to validate. So we had to dilute the sample multiple times in order to get a reliable signal. And if we used that with a less sensitive platform, we simply would have just lost the signal. But we did keep that signal here with the Simoa platform. So in my lab and multiple labs that I collaborate with, we use the Homebrew option on Quanterix for multiple different proteins. We've used it for neuropentraxin because we saw that there was a need for there to be a more accessible assay. And that means we could take this kind of research question into our own hands. We could generate our own antibodies, or we could purchase commercially available antibodies and build an assay and develop it ourselves. So we've now been able to create our own research tool in neuropentraxin-2, and we've done it for other biomarkers. And it gives us the flexibility and the kind of independence to do our own research.